Thank you, Nancy, and thanks to Shalev and Arjan for the opportunity to share some of our work here today. Among this rather star-studded lineup of speakers and leaders in the field, I might add. Although we're not exactly enjoying the slopes as we initially intended, I think when we got the invitation a few years ago, I definitely do hope that everyone is staying healthy and safe from the comforts of wherever you're joining us from. So let's see. Right, before I jump into things, I just wanted to first give a shout out to my team members. We are a reasonably new lab starting in the midst of the pandemic a little over half a year ago now. So we're definitely still recruiting and looking to establish new collaborations, particularly with more experimental labs. So if you're interested, please do reach out and we can get a conversation started. So I think it's well appreciated by this particular audience and highlighted by a number of talks recently or already, for example, by Michael, Itai, Shlev, Long, and a number of other speakers already, that the advances in sequencing and imaging technologies have recently really enabled this high throughput spatially resolved transcriptomic profiling at single cell and near single cell resolutions in fixed tissues. And here I'm just highlighting a few different technologies on the tissues that they've been applied to. And of course, there's many more, but it's hard to fit them all into the same slide. But briefly, these technologies allow us to measure not only what genes are expressed within these cells, but also preserve how these cells are organized within the tissue. So of course, given this very large and high dimensional data, we need, for example, new computational and statistical approaches to analyze them and extract out the relevant biological insights. So for more on our thoughts regarding some of these computational challenges in particular, and in particular, I guess, as we apply these tools and technologies to disease settings, please do check out our review in Nature Experimental and Molecular Medicine. Well, one of these technologies pioneered in Xiaowei Zhuang's group, where I conducted my postdoc research, is called multiplexed error-robust fluorescence in situ hybridization, or MERFISH. And what MERFISH does is it uses combinatorial labeling, error-robust barcoding, and sequential imaging to achieve spatially resolved genome-wide now transcriptome imaging with a very high, near 80% detection efficiency and a very low 4% error rate. But, given this very high detection efficiency and low error rate, it allows us to begin asking some interesting biological questions. So in particular, in our PNAS paper from 2019, we applied MERFISH to image 10,000 different, unique gene species and began asking questions related to the enrichment of different genes and different subcellular compartments, for example, exploring how mRNAs are organized within the cells. So we can, for example, combine MERFISH with ER staining and ask, what genes are spatially co-localized on a pixel level with the endoplasmic reticulum, and essentially test on a statistical level what genes are enriched in the ER versus outside the ER. And being that this is an imaging-based approach, it's also very nice that we can just essentially visualize the results. So we can visualize here each red point is a ER-enriched gene species that we identified, overlaid onto the ER staining image. And visually, we can just begin appreciating how the different ER-enriched genes are indeed spatially co-localized with the rough ER, even in the very fine mesh networks of the rough ER here. And likewise, we can, of course, check individual genes and so forth. But zooming out, we can also, for example, look at cellular organization and ask, for cells of a certain transcriptional state, if we look among its K nearest spatial neighbors, do we see any enrichment of particular cell types and cell states? And in this particular case, we were looking at, U2OS cultured cells that comprised 5 different transcriptionally distinct cell states. And indeed, cells in cell state 1 tended to be spatially co-localized with other cells in cell state 1. And again, we can just visually appreciate this in this plot here, where each point is a single cell and they've been colored based on their transcriptionally distinct clusters. And visually, we can see how cells of similar transcriptional states tend to cluster together with each other in culture. But of course, we can also apply these technologies to other cell types and even tissues. So more recently, this is work led by Guiping Wang, a very talented graduate student who is now pursuing a postdoc in Howard Chan's group. But she applied MERFISH to image and characterize the spatial organization of, I believe, over 4,000 different genes within cultured hippocampal neurons. And what this allows us to do is just begin really visually interrogating and also statistically interrogating the different spatial organization of different RNAs in, for example, the dendrites or more distal dendrites within the axons and other possible subdendritic and subcellular compartments of these highly polarized cell types. For more information on this, I do encourage you to check out her preprint that's available on bioRxiv. And of course, this data is very large, and so just exploring it can become a bit challenging. So we actually built on advances in video game graphics and rendering to develop an interactive web tool that allows us to explore, these millions of different mRNAs and their spatial organization. In this case, this is the neuron data set again, where, for example, each point is a different mRNA, and they've been colored based on species. So just visually, you can begin appreciating how, we have GAT2 and some inhibitory neurons here, and CAMP-K2A is more spatially colocalized to the distal dendrites, whereas KIF2C in blue is more colocalized with the axons, for example. But the story I really want to teach you all about today is actually in regards to a particular limitation of these approaches, and it's that these cells are fixed. So, they're dead. There's no way for us to follow them as they do, interesting things. And ultimately, the expression measurements that we obtain represent static snapshots in time. So even though, these measurements represent static snapshots, it doesn't mean that all temporal information is lost. And we can see that concept here where we have, the static snapshot of friends jumping off a cliff. But we're all smart enough to be able to infer in this particular case a pseudotemporal ordering regarding which friend must have jumped off the cliff first. And actually, with a few additional assumptions, such as how gravity goes down, we can actually provide a direction to the pseudotemporal ordering. Now we know that this friend must have jumped off before this friend, and so forth. And actually, given the information regarding the positions of these friends along this dynamic process, we can actually also begin predicting the future positional states of the people on top of the cliff at a future time point. They must be more like the next friend. Even though gravity goes down, we don't expect them to just plummet downwards. There's some information regarding angular momentum that we can infer based on the spatial positioning, in this case, of all the other friends. So this is kind of the same intuition behind RNA velocity analysis, where we can take advantage of the spatial patterns, in this case, in this static snapshot to make inferences regarding the future transcriptional states of these cells. And in particular, for RNA velocity in situ analysis, we'll leverage the nuclear and cytoplasmic expression levels to predict the future transcriptional state of these cells. Under these very simple assumptions that, if we recall from Biology 101, that mRNAs are transcribed in the nucleus, which we can observe from our static snapshot, and they're exported into the cytoplasm, which again, we can quantify and observe for each gene and each cell in our population. And presumably these mRNAs are eventually, translated into proteins and degraded and so forth. But that means, given this very simple model, well, we can model the cytoplasmic mRNA expression level, or the rate of change of the cytoplasmic mRNA expression level, as a function of what's coming in minus what's going out. And this means that at a certain steady state where, this rate of change is zero, we would expect there to be a certain ratio of the nuclear and cytoplasmic expression levels for a particular gene. And that means that when we look across a population of cells, If we, for example, see that some cell has more nuclear expression than what we'd expect in steady state, then we can interpret this cell as upregulating this gene, since there's more nuclear expression, there's more nascent transcription, and so forth. And alternatively, if a cell has lower nuclear expression relative to cytoplasmic expression compared to what we expect in steady state, then we would interpret this as this cell downregulating this gene. And indeed, when we look at real data, again, x-axis is cytoplasmic expression, and y-axis is nuclear expression for these particular genes. MCM6 in this example, we can see some cells, with higher nuclear expression and some cells with lower nuclear expression relative to cytoplasmic expression. And likewise for this different gene, KIF2C. So it seems that our model is appropriate for our data, and our data likewise adheres to some of the assumptions or the patterns that we expect to see under our model. So what this means is that, for example, for a particular cell, we have this high dimensional observed transcriptional state. In this case, for a particular cell, we have some genes that are lowly expressed in blue and some genes are highly expressed in red. But in addition to this observed transcriptional state, we can then use our RNA velocity models to predict the future transcriptional state of this cell. So for example, perhaps, here there's a gene in the middle that's kind of lowly expressed in the cell, but perhaps this cell, has higher nuclear expression relative to cytoplasmic expression. So in the future, we predict this gene to become more upregulated and turn red, for example. So what this means, of course, is that we can still do all our clustering analysis. We can take, for example, our matrix of genes by cells, and we can do our clustering analysis to project this data down to 2 dimensions using two-stochastic neighbor embedding or other graph-based embedding and dimensionality reduction approaches. And we can also identify transcriptionally distinct clusters of cells. So this is actually U2OS cultured cells, where, again, each point here is a cell, and the cells are closer to each other in Disney space if they're more similar transcriptionally, and they've been colored based on the graph-based clustering. So there are approximately 5 transcriptionally distinct clusters here. And again, we can do, differential expression analysis and find that MCM6 is most upregulated in the C2 cells, and KIF2C is most upregulated in this C5 cells, and also visualize the expression of these genes in the Disney plots, with red being high expression and blue being low expression. But of course, in addition to all this, as we would typically do, what RNA velocity analysis, as I noted previously, is because we have both the observed state and now we can predict the future transcriptional state, we can actually also project that predicted future transcriptional state onto this 2D embedding as well. And in this particular case, we can represent this predicted future movement in this lower dimensional transcriptional or lower dimensional embedding of arrows. And what we can just appreciate is that in this particular case, it goes in a circle. And perhaps if you're familiar with some of the genes that I mentioned previously, it's unsurprising that these would actually reflect the progression of these cells through the cell cycle. So we can actually take the angle along this circle here as the putative pseudotime and begin looking at the expression of different genes as a function of pseudotime itself. With, for example, MCM6, as we saw previously, was upregulated and then it becomes downregulated, versus KIF2C has a bit of an opposite pattern. But we can actually do this. We can then, look across all the genes that we've evaluated and test, on a statistical level, what genes have a significant proportion of their variance, for example, that can be explained by pseudotime, and in this manner identify, 1,000 different genes with transcriptional variance that seems to be associated with the cell cycle, so that we can, in this particular visualization, begin appreciating the gradual transcriptional changes that the cell has to go through in order to, as it progresses through the cell cycle. And of course, we can also apply these RNA velocity approaches to more complex systems, such as, in this case, neurodevelopment. And just visually, we can, in a very unbiased manner, for example, identify, in this case, the neuroprogenitor subpopulations that's giving rise to the different glial populations, and alternatively, differentiating into the neurons, as well as begin teasing apart some of the transcriptional dynamics that are occurring at these, early cell fate decisions, for example. So, however, the approaches that I've described so far for visualizing RNA velocities have relied on projecting, these observed and predicted future transcriptional states onto a 2D embedding that's actually made from the observed transcriptional state alone. So this can present challenges, particularly as we begin applying these approaches to more complex cellular trajectories, such as, for example, here we have a single-cell RNA sequencing data of pancreas differentiation, which is a system which is reasonably well characterized, and we expect there to be, for example, cycling ductal cells that give rise to endocrine progenitors that then give rise to the four terminally differentiated hormone-secreting cell types of the pancreas. So we can take the single-cell RNA-seq data and, again, do different types of dimensionality reductions, such as PCA, t-SNE, UMAP, diffusion maps, and so forth, and again, overlay our velocity arrows onto these 2D embeddings. But depending on which embedding we choose, we get slightly different, or at least we capture slightly different aspects of the underlying cellular dynamics. In particular, for example, maybe t-SNE and t-SNE and UMAP better capture the branching trajectory of the more terminally differentiated cell states, and also the cycling ductal progenitors, which is a feature we don't necessarily see, for example, in PCA. So, given this conundrum, what Lila Atta, a talented MD PhD student in my group, sought to do was actually create a 2D embedding directly from the RNA velocity itself. And she developed a software package called VeloViz to make this method accessible. And for more details of the method, you're definitely welcome to check out our preprint, which is on bioRxiv, as well as access some the software and tutorials on our lab website. But briefly, what this method does is it takes advantage of the current and predicted future transcriptional states from RNA velocity, and it computes this composite distance for all cell pairs that takes into consideration not only the transcriptional similarity between the predicted future transcriptional states of each cell and the observed cells in the population, but also the direction of that RNA velocity. Great. And then what this allows us to do is create a higher dimensional graph representation of each cell with every other cell, taking into consideration, these predicted future transcriptional states. And then we can, of course, prune certain edges of this graph based on other, high spreading distances or similarities. And of course, then visualize the resulting high-dimensional graph in 2D using various graph-based embedding approaches. And when we apply this VeloViz approach to pancreas development, again, what we see is that we can recapitulate the expected cycling ductal cells that differentiating into the endocrine progenitors, and then eventually give rise to the terminally differentiated hormone-secreting cell types, mirroring our expected underlying cellular trajectory. However, so far, the cellular trajectories I've shown you assume a reasonably uniform sampling of cells along this dynamic process. But what happens when intermediate states are missing? And this is a challenge that we've faced in some of our other collaborations, particularly in the disease setting, where we were concerned about certain cell populations being lost due to sensitivities and dissociation or other biological and technical challenges. And it's of concern to us because what we find is that a lot of these 2D embedding approaches that use only the observed information are able to capture, for example, these local relationships, in this case between friends, but it's not necessarily very good at bridging these gaps. And we can just simulate an example of this, for example, by removing, in this case, the pre-endocrine progenitors from this pancreas development single-cell RNA-seq data set. So we can remove, the pre-endocrine cell here and rerun all the velocity projections and 2D embedding. And expectedly, what we find, perhaps most best highlighted in this UMAP, is that even though these 2D embeddings are able to capture the local trends, in this case, capturing the cycling ductal cells and the progression into the EP cells, it doesn't necessarily preserve the global relationship across this particular simulated gap. And in this particular case, we might be led to an incorrect interpretation of the underlying biology, where we might naively assume that the endocrine progenitors are differentiating into something over here, and they're not at all connected to the terminally differentiated homosecreting cell types, for example. In contrast, in contrast with Velovis, because we are actually taking advantage and into consideration the predicted future transcriptional states, we are then able to essentially bridge this gap and connect these endocrine progenitors into towards these hormone-secreting terminally differentiated cell types and create a trajectory that's more consistent with the known underlying biology in spite of this simulated gap. And of course, we can do this also not only with the pancreas, but we can go back to the MERFISH U2OS cycling cells that I showed previously and just show that without Velovis, we can still recover this very nice circle of cells progressing through the cell cycle. But then we can also begin introducing a simulated gap, for example, by removing all the G2M cells and rerunning all this analysis and redoing the embeddings. And in this particular case, Velovis is able to still keep this cycling trajectory and relationship among the cells, but now, of course, we have a little gap where the G2M cells used to be before we removed them. Great. So then I hope I've been able to show you that with single cell and spatially resolved transcriptomic data, we really do need new computational and statistical tools to extract out relevant biological insights, as I'm sure many of the speakers in this lineup agree with me. But data from single cell and spatially resolved transcriptomic profiling technologies do represent, at least for now, static snapshots in time. So we can overcome this by using, to some extent, with computational modeling via RNA velocity analysis to predict the future transcriptional state of cells. And I hope I've been able to show you that by taking into consideration of these predicted future transcriptional states, we can create more stable representations of the underlying cellular trajectories. And with that, you're welcome to check out some of the tutorials and resources we have on our lab website if you're interested in trying out these analyses for yourself, or even just reproducing some of the results we have in our papers. You're definitely welcome to check out our lab's website at jeff.works. And with that, thanks to everyone again for your attention. This is my lab right now. This is our virtual lab holiday card this year since we weren't able to reconvene together. But with that, I'd also like to thank, in particular, my postdoc mentor, Xiaowei Zhuang, and some of the grad students, Chenlong Xia and George Emanuel in the lab, who generated the MERFISH data, as well as my PhD mentor, Peter Karchenko, and Ruslan Sodotov in the lab for his efforts in leading the original RNA velocity analysis. With that, please do feel free to reach out, and you can also find me now and then on Twitter.
Great, thanks. Thanks, Jane, for the wonderful talk. I'm just gonna go down and the list of questions. The first question from Jared Salzberg is, are you able to make any inferences about spatial rather than transcriptomic trajectories of cells, i.e. in a system such as the intestinal epithelium where cells undergo unknown translocation?
That is absolutely a really exciting question, and definitely something that we're looking into currently, whether we can not only predict the future transcriptional state of cells, but let's say take advantage of cells at different, migratory paths or, spatial locations within a dynamic, migratory process, and see if we can try to predict a potential migratory path of observed cells. So definitely a really exciting direction, and one that we're looking into and interested in collaborating with others.
Thanks, Jean. I want to ask my own question here. So you showed very nicely that you infer velocities not from intronic, non-spliced and spliced versions of the RNA, but rather from nuclear versus cytoplasmic levels. Can you comment on the advantages, disadvantages, and can you also use MERFISH by imaging intronic probes and exonic probes to get similar velocities?
Yeah, it's a great question. I guess for context for everyone else's benefit, the original RNA velocity analysis pioneered in the Karchenko lab with Stella Narson's group was based on single-cell RNA sequencing, where we leveraged the ratios of spliced and unspliced RNAs under the assumption that, RNAs are transcribed in an unspliced state, and then they get spliced and as they mature and get turned into protein. So it's the same logic where we can look at spliced and unspliced RNAs to look for, predict whether certain genes are being nascently transcribed and upregulated. The short answer is, in theory, yes, we should be able to, I guess if we had designed specific probes for, the introns of RNAs, and we could distinguish that from the fully spliced RNAs, perhaps by, using probes that span the exon junctions, I think it will be possible to do an imaging-based version of, the spliced and unspliced quantification. I do think that the nuclear and cytoplasmic, if we have imaging data, nuclear and cytoplasmic might be a more robust way of quantifying, these dynamics compared to spliced versus unspliced. For, I guess, a number of reasons related to how difficult it is to probe the dynamic range of unspliced RNAs by imaging. Since, I would expect them, as we've seen from Long Kai's lab's work, a lot of the nascent transcription happens at these small foci on the DNA, so it's hard to tell whether it's one gene or 10 genes, or it's all kind of just spatially aggregated there. It is interesting, though, and an ongoing project in the lab is actually to compare these two models and try to see if there's certain dynamics predicted from one that are not well reflected on the other. And likewise, as we move into disease systems, I think it's really interesting to see if, for example, RNA velocity predictions from in the disease setting might be, for example, more susceptible to alternative splicing or aberrant alternative splicing, but be a little bit more robust to the nuclear and cytoplasmic ratio quantifications, for example.
Thanks a lot, Jean. There are more questions, but we'll have to continue this on the discussion forum or the Slack channel.